Gillian is here today, and she will present her analysis now and gives you an idea of where we will go.
Okay, thank you very much, Aaron, for that very kind introduction. It's a huge privilege to be here speaking to you today. I'm going to have to apologise because this is going to be not only in English but with like a very thick, horrible Scottish accent. So I will try and speak slowly, and please bear with me. So the title of my talk today is Human History and Diversity Through the Lens of Genetics. And as Ernst has mentioned, I'm actually coming here from the Centre for Genomic Health at the Mount Sinai Hospital, which is situated in New York City. So I'm going to be talking about kind of human diversity and human history, but really with a focus as well on precision medicine, which is ultimately what we were interested in at Mount Sinai. So just to give you kind of a broad outline of what I'm going to be talking about today, I'm kind of partitioning the talk into two sections, where first I'm going to be talking about some of the research that we've been doing at Mount Sinai Hospital, which is using genetic data to begin to understand patterns of diversity and migration into New York. After that, I'm actually going to spend a little bit more time talking about methods for exploring genetic diversity and very, very fine-scale patterns of genetic diversity at the mega scale, even in kind of putatively homogeneous populations. So in particular, I'm going to be focusing on some work that we've been doing using the UK Biobank data. So as I'm sure you're all very aware, the story of genetic diversity and migration is a complex one. What you're looking at here is a figure from a review from 2012, which is really showing the kind of migration arcs of humans out of sub-Saharan Africa and then into Europe, Asia, the Americas and Australia over the past 100,000 years. So these historical migrations and the environments that our ancestors encountered throughout these migrations have really shaped the landscape of human diversity that we see today. So now we're in 2018, and in the past 100 years, migration has really continued to escalate at an unprecedented scale. And this has resulted in the genetic diversity landscape being changed in very radical and complex ways, both at a local level and at a global level. So we've kind of had a front seat to this rapid acceleration of migration in New York City, which is by kind of any metric one of the most diverse cities in the world today. I don't know if any of you here are familiar with the blog Humans of New York. So this is a kind of blog photographer who goes around and takes photos of just people walking about in New York City and asking for a story. And it gives you it's a very nice canvas for kind of understanding just that you're getting people who are coming from all across the world and from all walks of life. It's really just an apex of human diversity. So when we think about how to kind of quantify or measure human diversity, often in research people take what could be considered to be a very coarse grain approach. So one example is just to kind of ask people about their ethnicity using census labels. So what you're looking at up here is basically a kind of bird's eye view of New York. So these are the 5 boroughs like Manhattan, the Bronx, Queens, Brooklyn, and Staten Island. And here, different neighborhoods have been colored based on the kind of majority of individuals who are self-identifying as one particular ethnicity in the US Census category. So you can see already, when you actually look at the sort of map of New York City, just based on this granular sort of view of diversity, you're seeing that it's very structured, where you'll have particular neighborhoods that are enriched for a particular population of people who are self-identifying in a particular way. So we're working in a hospital and we're geneticists, and we wanted to understand whether we could kind of get a kind of a more fine-scale understanding of patterns of genetic diversity that we're seeing in New York City through our patient population. So in order to do this, we're using a resource from the Mount Sinai Hospital called the BioMe Biobank. And this is basically a patient-based biobank that's recruited throughout the Mount Sinai healthcare system, where if you're a patient and you walk into a Mount Sinai centre, people can ask you whether or not you would like to participate in the biobank. If you say yes, they take a sample of your blood. They ask you some survey questions, including things about lifestyle, where you're from, where your parents are from, and then they take that information and they take your DNA and then they anonymously link it to your electronic health records. So this is a resource that we've been working with for research purposes at Mount Sinai for both clinical and for population genetics kind of studies. So presently, we have about 32,000 individuals in BioMe that have all been genotyped on the same SNP chip, so that's the Illumina GSA array. These are people that are coming from all across New York City. When we start to look at the self-reporting information that they give about where they're from, where their parents and grandparents are from, what we learned is that we're actually capturing a very recent diaspora population. So only 65% of all participants in BioMe were actually born in the United States. And when we look at where they're reporting their parents and grandparents were born, we see that only 35% of individuals in BioMe report that all 4 of their grandparents were born in the United States, with many of them coming from basically anywhere else across the world. We actually have representation of over 130 countries in the world in our biobank, with a majority of individuals, especially coming from Caribbean countries like Puerto Rico and the Dominican Republic. So we have this genetic data, and we know that we're working with an incredibly diverse kind of genetic database here. So there's many different approaches that you can take in genetics to understand genetic diversity, but one approach actually exploits genealogical relationships and relatedness. But thinking about this not at a pedigree level, but at a population level. So the way that we do this is using a metric called identity by descent. So effectively, identity by descent describes tracts or haplotypes of the genome that individuals have co-inherited from a recent shared ancestor at some point in their recent genealogical history. So what you're looking at here are the chromosomes of 2 half-siblings who are sharing tracts of their genome identical by descent. If you look at this pedigree above, you can see that 1 generation ago, they have 1 shared ancestor, and then they have co-inherited these blue, these red segments identically from that ancestor. So because only 1 generation of meiosis, and thus 1 generation of recombination has separated these tracts, they're very, very long, and these 2 individuals are sharing long and abundant segments of their genome identical by descent. So that's because these are basically effectively first-degree relatives with only one generation separating them. However, these tracts of identity by descent can actually persist over many generations in individuals who have been separated by many generations, many meiosis, and thus are very distantly related, perhaps on the order of 5th or 6th cousins. In this example here, we're looking at identity by descent, or IBD sharing, between 2 5th cousins. So you can see here that because recombination has broken up these tracts over the generations, they're actually much shorter than in the example you saw before. However, they are persistent. They do persist in the population over many generations, and we are able to detect them using computational methods. So we can go out even further than 5th cousins and think about this at a broad population scale. When you're sampling many, many individuals in a population, you'll find that these tracts can have persisted for many, many generations. And the consequence of this is that you can have any 2 individuals, you can randomly sample 2 individuals from any population, and there is some probability that they will be sharing some of their genome IBD, so identical by descent, even if they perhaps don't know that they're related. So if I were to kind of randomly grab 2 people here and check in your genomes for IBD, we might find that you have some of your genome has been co-inherited identically from a recent shared ancestor that perhaps you would have no idea that you shared. Maybe it's 5 generations ago or 10 generations ago. So when we actually start to look at many, many individuals, we begin to see these patterns of what we call cryptic relatedness. So these are relationships that may not be documented in your genealogy or in your familial known history, but they persist in the genetic data and we're able to find them. And what's interesting is when you start to look at patterns of identity by descent sharing at a population scale, what you see is that the patterns that you will observe are highly influenced by various different demographic events that have happened in population history. So this includes what we call genetic bottlenecks, endogamy, migration, and admixture. So really how populations are moving and how populations are mixing are going to have a very profound influence on the distribution of patterns of IBD sharing that we see. And in addition to that, this idea of population bottlenecks, meaning that the number of ancestors that established a population, a contemporary population, is going to have a profound influence on the distribution of these IBD haplotypes that we see. So if very, very small number of ancestors established a population, that's what we would call a founder effect or a bottleneck. We will see very elevated sharing of these haplotypes because many individuals in the contemporary population share many ancestors at some point in the past. So when we start to explore these properties in contemporary populations that we've sampled, we can begin to make inferences about history and migration. So how do we find these segments? So we're using, in this case, genetic genotype data we call SNP chips. So basically we're sampling different polymorphisms at various different regions in individuals' chromosomes, and then what we're looking for are for segments that we're finding that are long and uninterrupted and completely identical. So the variants that we're looking at are polymorphic, so there's an expectation, a probabilistic expectation that if you have not inherited this identically from the same person, you will not see a long region which is completely identical between two different people. So that's how we're, that's how we're able to actually infer the presence of these IBD haplotypes across populations. So this is the method that we use. We use an algorithm called germline, which looks for these long tracts of the genome that are completely identical, and then we can begin to actually look at the properties of sharing between individuals in our database. So this plot here actually represents a way that we can start to visualize this in our data. So if you look at this plot, this is the biobank in New York, and every single dot here doesn't represent an individual, it represents a pairwise relationship between two individuals. So what we're looking at are the patterns and the distributions of IBD sharing between any given pair in our biobank. So there's two different ways that you can begin to think about quantifying IBD. One is just to look at how much of the genome you're sharing IBD in centimorgans. So this is what we have on the x-axis down here. Or we can look at how many IBD segments a given pair of individuals are sharing. So when we actually plot this for every pairwise relationship, what we see is we can pull out people who are directly related very easily because, as you remember from the first example, these individuals are sharing very, very long segments and very, very much of their genome is completely identical by descent. So this is these individuals up here who are kind of second, third degree relatives and above. And then when you look at this smear down here, this is kind of more akin to the pedigree of fifth cousins that we saw earlier, where you're not necessarily sharing an abundance of IBD, but there is the presence and the persistence of this IBD sharing. So we can begin to understand that there is a distribution of this that you see at a population level, which is representing the various different gradations of relatedness and kind of genealogical distance between any given pair of individuals in your data set. So that's one way of kind of thinking about and trying to quantify IBD. But another way is to think about kind of relatedness and IBD sharing as a network. It lends itself very well to network analysis because you're thinking about all these different relationships between all these different people at a population scale. So this is what we're looking at here, where every single dot here actually represents an individual this time. And they're basically kind of joined together by an invisible edge here, and that invisible edge is simply whether or not any given pair of individuals is sharing any of their genome IBD. So you'll see that some individuals are clustering very closely together, and then some individuals are much further dispersed from the rest of the network. So that's kind of a representation of how much these individuals are sharing and how related these individuals are to one another. So even just starting to kind of look at this network very naively, you can see that it's not completely random. You can see that there's some people who are clustering very tightly together, and then there's some people who are extremely dispersed. So we thought that was very interesting, and we wanted to know, first of all, whether this was something that correlated and was kind of demographically meaningful. Was this telling us anything about who these people were or where they were coming from? So as a first pass to begin to understand this, we simply decided to color the network based on people's self-identified ethnicities. So how are you self-identifying? So these are the US Census categories here and what you can identify as. And you can see already that there's kind of non-random patterns emerging in this network. So we have individuals who self-identify as African American over here in dark blue, tend to cluster more closely together. Then we see these clusters of individuals who are self-identifying as Hispanic or Latino in the middle, this light blue cluster here, and then this mix of red and light blue down here. It obviously appears to be non-random. So what we wanted to understand was whether by kind of applying some machine learning methods and applying more sophisticated computational approaches, could we actually pull out more information about who people are and where they're coming from, just by looking at these kind of genealogical relationships. So in order to do this, we borrowed a machine learning method from the kind of informatics sort of world, which is basically a community detection method. So the idea is, if you have a network and you want to understand whether parts of your network are enriched for sharing, or like for modules or modular, or whether a network is just completely random and everybody's kind of almost equally connected, you can apply this method to look for kind of regions of your graph that are enriched for sharing. So in our case, because we're looking at a network of relatedness, we're applying this method to try and understand whether there's regions of the subgraph that people are statistically enriched for being more related to one another than to the rest of the graph. So this is the method we applied. It's called InfoMap. It works basically by sending kind of just what we call a random walker around the network, and then if it gets stuck somewhere that's very densely connected, it'll spend a lot longer there, and then we can begin to make statistical inferences about, okay, well, these people are very, very related compared to everybody else. So that's basically what we're applying, and what it returns is what we call communities. So these are regions that show high modularity, where people tend to be much more densely related. So we applied this method to our network, and we were very encouraged to see that communities came out of individuals who were statistically enriched for having many shared genealogical relationships. One of the examples that popped out, it was one of the first communities to pop out, was this community in Burgundy here that were clustering very tightly together. But we actually looked at the people who were inside this community, who are the members. We found that 95% of the individuals in this community self-reported as being Ashkenazi Jewish. We applied some kind of complementary genetic ancestry analysis. Anyone who's done anything like 23andMe might be familiar with this. It's called principal component analysis. It's another way of looking at genetic ancestry. And we see that the people who fall into this community actually cluster together very tightly using this orthogonal method. So we're pretty confident that what we're pulling out is potentially demographically meaningful in this instance. It also makes a very kind of encouraging true positive, because anyone who's familiar with the Ashkenazi Jewish population may know that Ashkenazi Jews are considered to be a canonical founder population, which means that they've undergone a bottleneck at some point in their history and are thus enriched for IBD, basically. If there's elevated cryptic relatedness, as many individuals in the Ashkenazi Jewish community will have many shared common ancestors at some point in the recent past. So this was encouraging. When we saw this, we were very happy. We were actually able to pull out about 17 communities in total that had more than 100 individuals. And when we started to explore the self-reporting information that we had for these communities, we saw that these the communities that we were pulling out tended to be extremely highly correlated with kind of regional, continental, subcontinental, and even country level information that we had for some individuals. So these have kind of all been coloured based on community membership, and then what I've printed out to kind of annotate here is simply the best self-reporting label that we had for the community. And then the number below is a metric called the positive predictive value, which is just how good a job is this particular label doing of defining this community. So these are the various different communities that pop out. To kind of further validate this, we took a couple of examples. Remember, we're in New York City here. These are patients that are coming into our clinics, so we have information about where they're living and where they're coming from. So, for example, we took this very tight clustered green community, which we noted was very enriched for people who self-reported being born in Puerto Rico, and we looked at where they were living in New York City. And what we found was a very strong enrichment of people who were living in a neighbourhood called El Barrio, or East Harlem, in the southern Bronx. So I don't know how familiar people are with New York City here, but those are two kind of very well-known Puerto Rican neighbourhoods within the city. So again, it was very reassuring to see that this seemed to be recapitulating something meaningful. So there were many individuals that fell into this community who were born in the US and self-identified as Hispanic or Latino, but beyond that we knew nothing about their ancestry. And so when we took those individuals and we performed the same exercise, we saw the same pattern emerge. These people were living, again, in these Puerto Rican neighbourhoods. So we see them in El Barrio in the southern Bronx, and this is giving us kind of some confidence that what we're pulling out is truly kind of genetically defined shared ancestry that correlates very well with what we know about the patterning of the city. And it also proves that perhaps this is going to be a very useful method for understanding people's genetic ancestry in lieu of any self-reporting information. So in addition to kind of being able to explore where people are coming from in these communities, there's other very interesting properties that they have at a genetic level. So earlier on I told you that patterns of the distribution of sharing of IBD in a given population are informative for understanding patterns of migration, mixture, movement, and the presence of founder effects. So I mentioned that elevated IBD within a population is indicative of a founder effect. And when we started to look at the distributions of IBD sharing within the communities that we were detecting, we were not surprised to see elevated IBD sharing, for instance, in the Ashkenazi Jewish community, which is this one here in Burgundy. So as I mentioned already, this is what's considered to be a canonical founder population. They're very well studied. A lot is known about their demographic history, and it's not surprising that we see elevation of IBD within this community. However, when we started to look at some of these other populations, including the Puerto Rican community, the Dominican community, the Colombian community, so a lot of communities that are coming from various different places in the Caribbean and in Central America that are not very well studied, we see the same thing. We see elevation of IBD sharing, which is indicative that these may actually be founder populations. However, presently they are not recognised as such by the human genomics community. And there was one very interesting community that came out in particular, this Garifuna community here, which is this yellow kind of violin plot here, where on average any given pair of individuals within this community are sharing about 120 centimorgans of their genome identical by descent, which kind of puts them on the order of maybe being like fifth or sixth cousins. So it's a lot, it's abundant. So we're seeing these signatures of elevated relatedness, and we suspect that this may indicate that these populations underwent some kind of founder effect. So I mentioned earlier on that the length of IBD haplotypes decays as they're broken up by generations of meiosis, which means that very, very long tracts of IBD indicate very, very recent ancestry, while very short tracts indicate more distant and more ancient ancestry. So by taking populations and looking at the distribution of the length of the haplotypes of IBD within the population, we can start to actually model that in order to understand the effective size of the population over preceding generations. So this is what we're looking at here for two of our communities, the Puerto Rican community and the Garifuna community. We saw that they were elevated for having cryptic relatedness in the form of IBD sharing. So we ran these models, and what you're looking at here is on the x-axis, we're looking back into the past. So we're looking over preceding generations. And then on the y-axis, we're looking at how big the effective population size. So this is how many individuals were contributing to the kind of preceding generations at that given point in time. So this is all estimated from the length of the IBD haplotypes, and what we see is that in the Puerto Ricans, between 10 and 15 generations ago, we actually see a very, very profound population bottleneck, where the smallest effective population size of the contemporary Puerto Rican population was about 2,500 individuals, and that was about 12 generations ago. And anyone who's familiar with the history of Puerto Rico may know that this was around about the time of contact with Europe, which actually caused kind of a lot of complex demographic events to take place on the island of Puerto Rico, including the decimation of the native population, as well as this founder effect that's coming from the Iberian population that's moved in. So the numbers that we're seeing here are actually very congruent with historical reports of historical events that happened in Puerto Rico at the time. By contrast, if we look over here at the Garifuna community, we see about 11 generations ago an even more profound bottleneck with an even smaller effective population size of only 340 people. So before I started studying this population, before I was working in the biobank, I had never heard of the Garifuna before, so this took a lot of reading to understand who it was we were looking at here. So to give you a little bit of background, the Garifuna are a kind of an admixed Hispanic or Latino population that have on average about 80% African ancestry and 20% Native American ancestry. They're quite unique for a Hispanic population from the Americas, as they don't ever actually seem to have any European ancestry. And they seem to be extremely cryptically related. And we've seen that they've undergone this very profound bottleneck. And when you start to read into the history of the Garifuna, it's alleged that they are descendants of survivors of a shipwreck that was full of slaves that were coming from West Africa in the mid-1700s that crashed off the coast of St. Thomas. The individuals who survived stayed on the island of St. Thomas and integrated with the native Carib population there for many generations before eventually being displaced by Europeans to Central America. So they then lived in kind of isolated communities in Central America, where many of them still live today. However, there have been kind of more modern diaspora movements of the Garifuna. One such was to New York City, where there's a large Garifuna community now living in central Harlem. So we believe that this is who we're picking up here. So obviously we're actually, we're working in a hospital and we're a medical centre, and we're kind of really interested in understanding these kind of historical events and the presence of founder effects because they have important clinical utility. Basically, from a genetic perspective and from a clinical perspective, the kind of bottlenecking events that cause these kinds of patterns also can cause disease-causing mutations to drift to very high frequencies within these given populations. So you may have heard of examples of this before, like a very famous example is in Ashkenazi Jewish populations, the elevated frequency of BRCA1 and BRCA2 mutations, which can cause breast and ovarian cancer. So this is kind of a very classic example. So when we saw this profound bottleneck event that happened in the Garifuna population, what we wanted to understand was whether we could detect any variants that were known to be disease-causing, that were segregating at a high frequency here. So actually, we kind of did an analysis where we surveyed all the clinical variants that we had access to on our SNP array, and then looked at what was segregating. And in fact, we actually found one that was already known. So this variant is in a gene called MYPPC3. It's a pathogenic variant. It's in all the clinical databases, and it's causal for a disease called hypertrophic cardiomyopathy, which can potentially be a very fatal, very severe heart condition that you can actually live with for a long time without knowing you have it until you have a sudden cardiac arrest. So it actually kind of can contribute to quite a serious health burden. And what we found was this variant, again, is highly pathogenic, so it's extremely rare globally. And that's actually what we saw when we looked at it across the whole of our biobank. So then this plot here represents various different kind of population labels. So when we looked across BioMe, we saw that it was indeed very rare. It's even very rare amongst individuals who self-identify as Hispanic or Latino. But then when we look just within our Garifuna community that we're pulling out using this IBD network, what we see is that this variant is actually segregating above 2% in this population. So again, this is an autosomal dominant variant that's causing something that's severely pathogenic. And this variant has only been able to segregate to this frequency because of the demographic history of the population that we're looking in. So that actually wraps up the first section, and I'm going to move on now to talk about using similar methods in order to understand much more fine-scale patterns of genealogy and relatedness in a population that people would consider to be kind of putatively homogenous. So kind of a stark contrast to what we were just looking at in New York. So this is work that we've been doing using the UK Biobank data. I'm sure many of you are familiar with the UK Biobank. It's kind of a kind of open resource for researchers to do genetic data. It's very similar to the biobank I was just talking about in the previous section, where we have genotype data which is de-identified and linked to electronic health records. One of the big differences between what I was talking about before and this database is the size. So before I was talking about doing research in 30,000 individuals. I'm now talking about doing genetic research in 500,000. So of the 500,000 individuals who are genotyped in the UK Biobank, about 90% of those are self-identified white and are born somewhere in the United Kingdom. However, the other 10% are actually born in over 180 countries in the world. So that comprises about 50,000 individuals, actually making this one of the largest and most useful resources for studying genetic diversity that's kind of presently out there for the human genomics community. So again, we now have many more possible pairs of individuals who could be sharing IBD. So when we applied the methods that we were applying before to this data set, it was a lot more computationally intensive. We actually had to write an entirely new algorithm to do it. And eventually we found over 14 billion tracts of IBD sharing were present within this kind of 500,000 people. So there's a lot of cryptic relatedness in there, even though this is not a founder population by kind of any of the kind of conventional definitions. So when you start to just look at very kind of coarse grain kind of IBD sharing distributions, so just stratified by the 4 countries in the UK. So these are kind of very similar to the plots that I was showing earlier, where we're just looking at the distribution of how much IBD sharing any given pair of individuals is sharing within a country in this case. We don't see any kind of very striking patterns jump out. Most people aren't sharing a lot of IBD. They're very exponentially distributed, and when people do share IBD, they tend not to be sharing a lot. So just very, very coarse-grained bird's eye view, perhaps not the most interesting. However, we can start to look at some very interesting patterns of demographic history using this IBD anyway. So what you're looking at here is actually not IBD sharing just within the UK, but how much IBD various different regions in the UK are sharing, in this case with Northern Ireland. So this is kind of a way of kind of understanding how populations mix and what IBD can tell us about that. So what you can see here is basically the green means that there's not a lot of IBD sharing between a given region and, in this case, Ireland. The brown means that there's actually elevated IBD sharing. So that's kind of how to interpret the heat map here. And what you can see is an enrichment of IBD sharing in the west coast of Scotland with Northern Ireland. And if people who are familiar with the kind of the history of kind of mixture and movement between these 2 regions, that shouldn't come as a surprise at all. That's something that's very well historically documented. We can start to get some interesting insights though into more ancient admixture with the United Kingdom. So this is the same plot again, except this time what we're comparing is sharing of IBDs, the sharing of recent common ancestors between various different regions in the UK and, or in this case, it's Norway. So on the right-hand side here is the heat map of IBD sharing. You'll see that we see an enrichment of IBD sharing between Shetland, Orkney, and the Outer Hebrides in particular with Norway. And then on the left-hand side here, what we have actually is a map of the trajectory of the Norwegian Vikings down past Shetland and Orkney and then down the northwest coast of Scotland. So what we believe we're picking up here are perhaps very ancient signatures of admixture pulses in the form of recent shared ancestry between these populations. And then by contrast, if we look at the fraction of IBD sharing between various different regions in the UK and Denmark, we see basically the other part of the map lights up. Instead of seeing an enrichment within northwest Scotland, we're now seeing an enrichment in central England. And then if you refer back to the map over here, with Viking migrations from Denmark to the United Kingdom, we see that these patterns actually seem to recapitulate that effectively, and that's what we believe that we're likely to be picking up here. So in addition to kind of looking at these patterns of admixture, we can actually also think about founder effects, but this time not thinking about things at a population level, but thinking about things at a regional level. So what we're looking at in this plot is, again, it's just IBD sharing. So we were talking about this, how we quantified that Puerto Ricans were a founder population and the Garifuna were a founder population by looking at the presence and abundance of IBD. So we're looking at this at kind of a geographical kind of level at this point, and we can't really define these into discrete populations. But when we just begin to look at where people are living within the UK, we see that there are kind of very, very fine-scale patterns of differences of IBD sharing, even just within this one country. So we see highly elevated IBD sharing in the islands and the traditionally more rural areas. So the Outer Hebrides, again, Shetland and Orkney really light up as being very enriched for IBD sharing. Whereas we see extremely low levels in regions of Greater London. And this kind of makes sense based on what we know about the demographic history of these places, that places that are historically more rural and more isolated and less subjected to kind of gene flow from other places are going to have elevated relatedness relative to the more cosmopolitan areas that are going to be more subject to kind of movement and admixture and migration, where we tend to see less relatedness and less IBD. So because again, we're kind of interested in this from a personalised medicine perspective, understanding that we see these signatures of cryptic relatedness, it kind of opens up the question as to whether or not there could be disease kind of causing alleles or kind of a genetic burden that's specific to places that are enriched for having this shared genealogy, as we saw with the Garifuna earlier. So this is something we've begun to explore, and one of the questions we first wanted to ask was, well, do we see enrichment of particular diseases within particular regions of the UK? Again, we're working with a biobank here, so in addition to having information about ancestry and about genetic data, we also have health outcomes. So in this case, we're just looking at ICD-10 codes, which are basically just billing codes, but we can use those as a proxy for diagnoses in these biobanks. So when we start to look at whether or not there's an enrichment of these within given regions, we find, for instance, in the Outer Hebrides, we see an enrichment of endocarditis. In Orkney, we saw an enrichment of mucin metabolism disorders. And then in Anglesey, which is in North Wales, we saw primary ovarian failure. So it seems as though not only is there a very fine-scale genetic structure within the UK Biobank, there's also interesting substructure in the phenomic space as well, which I think is very important implications for how we think about setting up these kinds of study designs. So to conclude, basically we can use genetic data, as I hope I've shown you today, to reconstruct patterns of recent human demography. Founder effects in human populations are not always discrete. They can represent a continuum of cryptic relatedness, which is a lot like what we saw when we looked at the levels of IBD sharing within the kind of islands and rural regions of the UK. We can use identity by descent and network-based analysis in order to really begin to dissect and pull apart these large-scale databases at a very, very fine-scale granular level. And I think that this has huge implications for how we think about genomic medicine and how we conduct this research. And with that, I would just like to say a huge thank you to my collaborators, and thank you very much everybody for your time today, and I'd be more than happy to take any questions.